Hello everybody, welcome back to another edition of Ed Puzzle Lecture Notes. We are continuing on in nervous tissue. This is our third lecture on nervous tissue. And today we're going to go over the neuroglial cells. So last time we learned about the neuron. That's like the star of the show, the neurons. Well, these are the little guys like behind the scenes which support the neurons. They do a lot of jobs to help support the neurons so they can function properly. And here are some things you're going to have to know. They make up a large portion of the CNS, or the central nervous system, which make up about half the volume. They're smaller than neurons. Do know this. They are much, much more numerous than neurons. So there's a lot more little helpers, kind of like Santa. He has a lot of elves. One Santa. Takes a lot of elves to support Santa. Same thing with the neuroglial cells. Much more numerous. We have to know exact? No, because look, we don't even know exact. 5 to 50 times more numerous? Just know they're more numerous. Now these cells can divide. Remember, neurons are amitotic, so they cannot reproduce, but these guys can. We're going to learn basically 4 different types, and we're going to go over them coming up here in detail. We have the astrocytes, oligodendrocytes, microglia cells, and ependymal cells. Now those 4 are the ones that are active in the central nervous system, that is the brain and spinal cord, and then 2 which are active in the peripheral nervous system, all of your neurons located outside of the brain and spinal cord. Those are the Schwann cells, which we've kind of gone over a little bit, and also satellite cells. Now I know we went over these. The Schwann cells, those are the one that wrap the axons of the neurons in the peripheral nervous system with the myelin sheath. I don't remember seeing the role of satellite cells, but these guys are kind of similar. They actually enclose not the axon of the neuron, but they actually make like a sheath around the cell bodies of neurons in the peripheral nervous system, and kind of help to regulate the environment or the conditions around the cell bodies in the peripheral nervous system. And our first little helper cell for the neurons are the astrocytes. And they kind of look like little stars. So when I think of astro, I think of astronomy, and I kind of start thinking of stars. Because yes, they are star-shaped cells. So, things you need to know. Number 1, it's in red for a reason. They help to form the blood-brain barrier they have these little end feet. And they kind of enclose the capillaries which travel around the brain. Now, it's important that there is a blood-brain barrier because, of course, the brain is pretty important. You only want certain things to freely flow into the brain. So this barrier allows gases like oxygen and CO2 to freely flow through. Also, lipid-soluble substances can pass through, but it blocks others. Now this is good because they can block things like bacteria from getting into the brain, which obviously would not be good. These guys are also like little cleaners because they metabolize or break down the neurotransmitters that are released, and they also clean neural debris. They regulate chemical balance for the nerve impulses to occur, and they play a role in repairing any damage. The next one, the longest word, is the oligodendrocytes. What you need to know is basically these are kind of like the Schwann cells of the peripheral, that are in the peripheral nervous system, but these guys are in the central nervous system. So these are the guys that are going to wrap the myelin sheath around the axons of neurons in the CNS. So that's one difference between Schwann cells and oligodendrocytes. Oligodendrocytes work in the CNS. Another difference is that oligodendrocytes can wrap several neurons, where one Schwann cell, so like if this is the PNS, this would be one Schwann cell. It could only wrap a portion of one axon, where oligodendrocytes can actually reach out and wrap several different neurons. So they can put the myelin sheath around several different neurons, not just one, like the Schwann cells do. And remember that myelin sheath is important because it speeds up the electrical impulse transmission. It speeds up the rate of the action potential down the axon because remember, it allows it to jump from node of Ranvier to node of Ranvier. Microglial cells, or the microglia. Small cells found near the blood vessels. They play a phagocytic role, which means they like to eat stuff, like this guy right here. He's going to eat some sort of cellular debris. So he's like another cleaning crew. So you're probably saying, hey, that's kind of like the astrocyte. You're right. So when it comes to the astrocyte, not the microglial cells, remember the blood-brain barrier, because that is very important for the astrocytes. So the microglia is similar, but these guys do not help form the blood-brain barrier. These guys are more like just a cleaning crew for your neurons. Ependymal cells, very important. So you can see these guys are simple because it's just a single row. They're not stacked upon each other. They're columnar in shape. You can see kind of like a column. They're also ciliated. They have these little cilia, almost like hair-like structures on the inside. Now these guys line the ventricles of the brain and the spinal cord. The ventricles are like little canals, and through the canals, it's not water that flows through there, but it's cerebrospinal fluid. So these ependymal cells, they help to both produce, and with their little cilia flapping around, they also help to circulate the cerebrospinal fluid through the ventricles. And this is important because the CSF helps protect the brain, and it's also the way the brain gets its nutrients delivered from the blood. So that cerebrospinal fluid, the CSF, needs to be continually circulated around through the ventricles of the brain and the spinal cord. Now we are in the peripheral nervous system. And we've kind of gone over it already, but Schwann cells. So these are the guys that if there's an axon like this, they're what produce the myelin sheath. And remember, each individual little part of the myelin sheath, that is a single Schwann cell like this right here. So basically you just need to know these guys are what produce the myelin sheath around the axons of neurons in the peripheral nervous system. Also remember, these guys can only work on one neuron. Each cell can only be a part of one myelin sheath in one neuron, where the oligodendrocytes can wrap the sheath around multiple neurons. And also remember that the myelin sheath is important because it allows the nerve impulse to jump from node to node to node of Ranvier, which speeds up the action potential much, much faster. And remember that that is also called saltatory conduction. So when it jumps from node to node to node, that's called saltatory conduction. And you're probably tired of hearing about it, but hey, here's myelin again. That's the substance that surrounds the axons of neurons. Remember, Schwann cells do it in the PNS, peripheral nervous system. Oligodendrocytes do it in the central nervous system. A lot of time we'll just use those three letters because it gets old to keep writing it all of that. These things are made up of mostly lipids, 80% lipids. That's why it appears white. Lipid is fat, basically. And 20% protein. And the main thing that you need to know is that it increases the speed of the electrical impulse as it travels down the axon of the neuron. And another little role is that since it's kind of insulated, it prevents the impulse from leaving the axon and possibly stimulating a nearby neuron. Myelination in the peripheral nervous system, remember that's done by Schwann cells. Remember these guys can only work on one axon, one neuron. The Schwann cells, they cannot myelinate several axons like the oligodendrocytes. Let's see, most of this stuff you're not going to need to know, I don't think. The neurolemma part is this part right out here. It's the very outside, which includes the cytoplasm and the nucleus of the Schwann cell. And this in here, this section right here would be the myelin. Don't remember that being on our tests. Remember the nodes of Ranvier, those are the gaps that the nerve impulse jumps from gap to gap to gap, which helps speed up the nerve impulse. And I'm fairly certain you don't need to know it, but the Schwann cells myelinate or wrap around the axons of the peripheral nervous system during fetal development before you're born. And that's basically what you need to know about Schwann cells. Just remember they work on one neuron, not multiple neurons, and they work in the PNS, peripheral nervous system. Those are the two main things you need to know. And a little diagram here. I'm not going to go much on this because I don't see much in here that you need to know. Of course, here is the axon in here. You won't need to know it, but the axoplasm, that is the very inside of the axon. The axolemma is the sheath or the very, very outside membrane around the axon. Don't need to know that, so don't write it. But just remember, this is what the myelin sheath looks like. That would be a Schwann cell. These would be individual Schwann cells. This, of course, is a node of Ranvier. So the nerve impulse, since this is myelinated saltatory conduction, it would jump from node to node to node, making the nerve impulse or action potential travel much faster. And myelination process in the central nervous system, the CNS, remember that's done by oligodendrocytes. You can see one right here. Now they do it a little differently. They don't wrap themselves around the axon like the Schwann cells do. They have these processes which come out, and you can see this guy's reaching out and myelinating this portion, but he's also reaching out here and myelinating another portion of that axon. Remember, Schwann cells cannot do that. Each one of these in the PNS would be an individual Schwann cells, where oligodendrocytes can reach out with their processes and myelinate several different parts of the axon or different axons on different neurons. It says no neurolemma is formed. That is true because the cell does not wrap itself. So this guy does not come in there and actually wrap his cytoplasm and the nucleus around the cell. It does it through its processes that reach out. And therefore, little regrowth after injury is possible when it comes to the CNS. But to be honest, what you need to know, oligodendrocytes, they're what myelinate the axons in the CNS, and also know that these guys can myelinate multiple axons, not just one like the Schwann cells do in the PNS. All right, last slide, some key terms. First one is called a ganglia. Ganglia, those are clusters of cell bodies, the cell bodies of neurons, so a bunch of cell bodies clustered together outside of the brain and spinal cord. So they're in the PNS, peripheral nervous system. So a bunch of, or a cluster of cell bodies, not axons, but cell bodies, outside of the brain and spinal cord, that's called a ganglia. Now, that same thing inside the central nervous system, inside the brain and spinal cord is called a nucleus. So that's the same thing. It's a cluster of cell bodies inside the CNS. It's called a nucleus. Now you probably heard about it. It's called gray matter. You hear gray matter and white matter. The white matter, those are axons, and those are myelinated axons. They look white because they have myelin wrapped around the axon, and myelin is made out of mostly fat, and that appears white. The next term I'm sure you heard of, it's called a nerve. Now these sensory and motor neurons, they don't work alone. They work in bundles of neurons, which also has some connective tissue and also blood vessels to help support all the neurons. But these bundles of neurons together form a nerve. And the last one is a tract. And that's a bundle of axons in the central nervous system. So a bundle or a lot of axons together in the central nervous system makes up a tract. And these tracts help to transport sensory information from the body to the brain and spinal cord, and also connect neurons in the brain and spinal cord. All right, thank you very much for listening, and I'll talk to you next time.